Bacteria and microbiological hazards. Introduction. Perhaps the most prevalent and tenacious hazard in food safety is food poisoning, the primary cause of which is microbiological contamination. Food poisoning can be caused by bacteria, viruses, or molds entering the food and spreading illnesses or diseases or introducing toxins to the food. In this module, you will discover these microscopic enemies and what they do. Later in the course, you will find out how to prevent them from making food harmful and what the law has to say about your responsibilities and obligations. Topics to be covered in this module: bacteria, types of bacteria, helpful bacteria, spoilage bacteria, pathogenic bacteria, sources and vehicles of bacterial contamination. How do bacteria multiply? What do bacteria need to multiply? Cross-contamination. Bacteria and the danger zone. How to prevent bacterial contamination. Viruses. Molds. High-risk and low-risk foods as biological hazards. Bacteria. In Module 1, we saw that the greatest danger involved in food handling is food poisoning. The biggest cause of such illness is from bacterial contamination. Bacteria are microscopic single-celled organisms which inhabit almost every place on Earth and can be found on and in almost every object, including food and food equipment. They are found from the deepest parts of the oceans to the upper reaches of the atmosphere and from the soil beneath our feet to the human gut. Most are harmless and many are beneficial to us, while some are harmful in the wrong place or cause food to spoil or spread disease. Types of bacteria. There are 3 kinds of bacteria for our purposes: helpful bacteria, spoilage bacteria, pathogenic bacteria. However, it is the last 2 which cause problems in food handling and production. Spoilage bacteria make food smell off, become discolored, and otherwise unpalatable as it decomposes. Pathogenic bacteria cause disease and illness. Types of bacteria. Pathogenic bacteria are usually undetectable by sight, smell, or taste when they are on food, but can multiply at such a rate that threatening quantities can still be present on even the freshest of foods. This is the reason that food standards laws and regulations were created, to remove the conditions in which such bacteria multiply and to prevent them from reaching humans and causing food poisoning. Helpful bacteria. Not all bacteria are harmful, and in fact, we wouldn't exist without some kinds of helpful bacteria. These are mostly found in dairy and fermented foodstuffs. Examples of foods which contain helpful bacteria include live yogurt, which includes probiotic bacteria; sauerkraut, fermented cabbage preserved in salt water; tempeh, fermented soybean extracts; kimchi, Korean cabbage fermented in spices; miso, fermented soybean paste; pickles. However, we now need to consider the kinds of bacteria that poison food. Spoilage bacteria. Food spoilage is essentially the state where food begins to decompose and is usually easy to spot. Spoilage will make the food unappetizing, and you should not consume food in such a state. The kinds of organisms which cause food spoilage eventually grow and reproduce on any food items left unprotected and include molds and other fungi, viruses, and certain types of bacteria which are called spoilage bacteria. However, these aren't necessarily dangerous but cause food to be inedible and unfit for human consumption. Pathogenic bacteria. Many significant types of bacteria cause the genuinely health-threatening set of illnesses known as food poisoning. These are the pathogenic bacteria, and they can be particularly dangerous because there are often no sensory indications of their presence in food. You come into contact with thousands of types of bacteria all the time. However, it is the common but harmful types that get into our food and water and cause illnesses that can sometimes be fatal. There are more than 1 million cases of food poisoning in the UK every year, which result in more than 20,000 hospital admissions and about 500 deaths. There are 7 frequently encountered major types of pathogenic bacteria which occur in our food. Salmonella, Clostridium perfringens, Bacillus cereus, Campylobacter, Escherichia coli, E. coli 0157, Listeria, Staphylococcus aureus. How do bacteria multiply? While they are mobile life forms, bacteria reproduce, grow, and multiply very differently from animals, by splitting in two. If there are enough nutrients in a bacterium's immediate environment, along with adequate temperature and moisture, it will divide exponentially. This will result in rapid multiplication in the space of quite a short time. Some kinds of bacteria can double in number every 20 minutes. This means that one bacterium can become more than 16 million bacteria in 8 hours. Clearly, controlling the conditions needed for bacterial growth is an essential part of any food handler's responsibility to ensure that bacteria don't get the chance to spread and become a danger to humans. What do bacteria need to multiply? The conditions needed by bacteria to multiply are perhaps best remembered using the acronym FATTOM, FATTOM. This stands for food, acidity, temperature, time, oxygen, moisture. FATTOM. FATTOM can be further explained by outlining how each of these factors encourages bacterial growth. Food. With warmth and moisture, protein-rich foods are particularly suitable for bacterial growth. Acidity. Vinegar and lemon juice are good preservatives because most bacteria can't survive or grow in acidic conditions. Temperature. The optimum temperatures for bacterial growth are 4 degrees Celsius to 60 degrees Celsius, a range known as the danger zone. Time. The longer foods present the conditions for bacterial growth, the more dangerous they become, as they create an increasingly favorable growth environment. Oxygen influences bacteria by presence or absence. There are 2 types reliant on these conditions. Aerobic bacteria need oxygen to multiply, can be prevented from growing by vacuum-sealed containers. Anaerobic bacteria only multiply with the absence of oxygen, will contaminate jars or cans of food which have been improperly processed. Moisture. Bacterial growth is faster in moist conditions, which is why dried and salted foods tend to be viewed as low risk. Sources and vehicles of bacterial contamination. There are many ways in which bacteria can enter food, and these can be grouped into 3 main routes: passive, indirect cross-contamination, and direct cross-contamination. We'll discuss cross-contamination in more detail below, but there are several forms of passive contamination routes. Food animals. Pathogens transferred during slaughter from their skins or intestines, with fecal matter crossing over during evisceration. Poultry. Slaughtering machinery can transfer bacteria from bird to bird, while Salmonella can get onto eggs. Shellfish. Bacteria accumulates via filter feeding in live mollusks, particularly risky if eaten raw. Raw fruit and vegetables can carry soil microorganisms from contamination by insects and other creatures via urine and walking on food. Milk must be pasteurized to kill bacteria, as milk is a perfect reproduction substrate for bacteria. Water must be properly treated to neutralize the runoff from soil, sewage, and slurry. Stagnant and storage tank water should only be used for washing, not drinking. Cross-contamination. There are two kinds of contamination: indirect, direct. Indirect contamination happens when pathogenic bacteria are passed from a source via a vehicle to the food. The following are examples of when this can occur: handling food without washing your hands or wearing protective gloves, not washing or disinfecting a chopping board or knife between use on raw meat, when followed by cooked meat or vegetables, using the same dishcloth to wipe raw food preparation areas as cooked food areas. Direct contamination happens when the source of the pathogen contacts food without an intermediate stage. Examples of this can include when the food handler coughs or sneezes onto the food, when raw chicken is stored above cooked chicken in a fridge, when fresh meat is stored among or next to cooked meat, when uncooked seafood is stored above salads or vegetables. Bacteria and the danger zone. As we have seen under FATM, warmth is one of the factors which encourages bacterial growth. Bacterial growth is particularly rapid in the danger zone of 4 degrees Celsius to 60 degrees Celsius. In fact, most bacteria can't reproduce outside this range. It is perhaps our misfortune or evolution's ploy that our healthy body temperature sits right in the middle of that range, at 37 degrees Celsius. This is the optimum reproduction temperature of most bacteria which cause sickness in humans. Consequently, food safety protocols and laws insist that hot food is stored above 63 degrees Celsius, while cold food is stored below 5 degrees Celsius so that bacterial growth is prevented. By law, you have to store cold food below 8 degrees Celsius, though best practice is below 5 degrees Celsius. All food served hot or cold must have been cooked thoroughly to kill bacteria. Undercooking can actually encourage bacteria to grow and reproduce. This means that you must cook food at 70 degrees Celsius or above for 2 minutes and preferably more. How to prevent bacterial contamination. The following rules will ensure that you prevent the growth of bacteria and prevent cross-contamination. Raw and ready-to-eat food should be prepared separately using cleaned and disinfected equipment. Fresh vegetables must be washed thoroughly in a designated separate sink. Utensils should never be washed in the same sink as raw food is prepared. Disposable single-use cloths should be used for wiping or drying utensils and work surfaces. You must wash your hands thoroughly before handling food, as well as after handling raw food. Food should be moved and put in place using tongs, spoons, or ladles wherever possible. Gloves should be used where possible and changed after each food handling task. You should wash your hands before wearing protective gloves. Never leave food standing in a warm or moist environment. Prepare food in the smallest amounts possible so that it isn't kept out of refrigeration for very long. Viruses. Viruses are also responsible for some food pathogens and are different organisms entirely to bacteria, though still microscopic. A virus is essentially a parasite which reproduces itself inside other organisms' cells, thus infecting them and causing diseases. However, because they need to use a living cell to do this, they don't multiply within food, but need to be ingested to reproduce. The two most frequently seen viral foodborne illnesses are norovirus and hepatitis A. Both can transfer between people easily and can survive for lengthy periods on door handles, light switches, and work surfaces. Both are common in shellfish and other kinds of seafood, which means that such foods must be certified as having been collected from approved locations. There is little protection from viruses other than by thoroughly cooking foods, as they can be disinfectant resistant and also survive bactericidal hand washes. Molds. Also microscopic, molds are forms of fungi which live on mostly dead animal or plant matter. They are transferred via their even tinier spores, which can be transported via the air, water, or living creatures such as flies. Despite being mostly visible as a green, blue, yellowish, or white surface dusting, it is far more extensive than can be generally seen, having thin roots which penetrate down below the surface of the food. It is not just unsightly. Molds exude enzymes to digest the food, giving rise to the telltale signs of spoilage or decomposition in it, such as a moldy or offensive smell, atypically slimy appearance, and unusual color. Not only that, but some kinds of mold also introduce toxic substances into the food, known as microtoxins, which are fatal. Typically, these are ingested via wheat or other grains, which have become moldy in storage. High-risk and low-risk foods. High-risk foods. Because of the conditions needed to encourage the multiplying of bacteria, viruses, or molds, certain foods are more likely to carry them. These are known as high-risk foods. Such foods are generally those which are ready to eat, and examples include the following: pre-cooked fish, poultry, and meat, including deli meats; shellfish and other seafood; cheese, cream, fresh milk, and other dairy products; stocks, gravies, sauces, chowders, and soups; cooked pasta; cooked rice; unwashed leafy greens and salads; unwashed fruit; vegetable sprouts. Low-risk foods. These are foods which usually don't need to be chilled or kept warm, and are often dry or preserved in some way. Consequently, they don't provide a favorable environment for microorganism growth. Common examples of such food usually have a best before or shelf life date on their packaging or labels and include crisps and corn chips, crackers and breadsticks, packeted breakfast cereals, dried rice, pasta, quinoa, and couscous, pastries, cakes, and biscuits without dairy-based cream or fillings, sweets and other sugar-based confectionery, canned drinks, tinned food, cooking sauces in sealed jars, condiments, including table sauces. Once some of these are opened and out of the refrigerator, they quickly become high-risk foods and should be stored and consumed as directed on their packaging. Summary. In this module, you have learned that there are 3 different types of bacteria in terms of their effect on humans: helpful, spoilage, and pathogenic. Bacteria, viruses, and molds all cause forms of food poisoning and need to be controlled to prevent this. Bacteria need certain conditions to grow, described by the mnemonic FATM: food, acidity, temperature, time, oxygen, and moisture. Food handlers need to protect against both direct and indirect cross-contamination by being scrupulously clean and hygienic. The bacterial growth danger zone is between 4 degrees Celsius and 60 degrees Celsius, and the optimum temperature is the human body temperature of 37 degrees Celsius. There are high-risk foods, which are those which are cooked, ready to eat, or unwashed, and low-risk foods, which are generally dried or preserved.